Hi everyone, we are in week 5 and this week's lesson is about water testing. Having potable water is crucial for us to survive, and potable means drinkable, and the water supply can become contaminated by animal and human activities. So if the water supply becomes contaminated, people can become very ill. Fortunately, public water systems take many steps to ensure that the public has safe, reliable drinking water. And one of the most important steps is to regularly test the water. Sometimes there can be intestinal contaminants of fecal origin. So that can happen from sewer overflows and wastewater releases getting into the water supply. And around 3.4 million deaths a year are caused by enteric pathogens, which means bacteria that cause intestinal diseases, and that happens especially in developing countries. Some examples of those diseases that can be spread by enteric pathogens in the water supply include dysentery, typhoid fever, and cholera. So it's really important to monitor the water supply. But testing drinking water for all those possible pathogens would be a really complex, time-consuming, and expensive process. So instead, water supplies are tested for the presence of indicator organisms, such as the most frequently used indicator species, coliform bacteria. And you may have heard of coliform bacteria before, but what are they? Well, coliform bacteria are organisms that are present in the environment and in the digestive tract, so in the feces of all warm-blooded animals and humans. And coliform bacteria will likely not cause illness, but their presence in the drinking water indicates that disease-causing organisms or pathogens could also be present in the water system. So now we're going to talk about total coliforms, fecal coliforms, and E. coli. So total coliforms is the most broad category. So the most basic test for bacterial contamination of a water supply is to test for the total coliform bacteria. And if you look at this figure here, it shows you the total coliform is the biggest circle, the most broad category. And by testing for that, the total coliform count gives a general indication of the sanitary conditions of a water supply. So total coliforms include bacteria that are found in the soil, in water that's been influenced by surface water, and in human or animal waste. And most coliforms are not considered pathogens. So this is the broadest group, is their total coliforms. So the total coliforms are the most general group, and then within that group, the fecal coliforms are the group of total coliforms that are considered to be present specifically in the gut and feces of warm-blooded animals. So because the origins of the fecal coliforms are more specific than the origins of those total coliforms, fecal coliforms are considered a more accurate indication of animal or human waste than the total coliforms. And then even more specific, E. coli is within the fecal coliform group. So E. coli is the major species in that fecal coliform group. Of the groups of bacteria that comprise the total coliforms, only E. coli is generally not found growing and reproducing in the environment. It's not an environmental species. So E. coli is considered to be the species of coliform bacteria that's the best indicator of fecal pollution and the best indicator of the possible presence of other pathogens. E. coli is a species of coliform bacteria, and they're always present in the intestines of warm-blooded animals, including humans. So E. coli is always present in our intestines. That doesn't mean we're always sick from it. Most strains of E. coli are harmless. But the fact that it's always present in our intestines makes it a good indicator species. So its presence in the water supply suggests possible fecal contamination. And again, most E. coli are harmless, but there are some strains that can cause illness. So aside from testing the water supply, which is generally very safe where we live, there are other applications for water testing. For example, testing lakes and bodies of water where people swim. So imagine a worker for the state health department is given the job of regularly analyzing lake water from a public recreation area. They would check for the presence of coliform bacteria because that can indicate fecal pollution. So they would need to ensure that the water contains no appreciable number of coliform bacteria that could potentially make people sick from after swimming or playing in the water. So that's what we're going to do this week. We're going to test some water samples from some different water sources and check them for total coliforms, and that's a screening method to look for possible contamination. So testing for coliforms is based on the facts that they are gram-negative bacteria, they're non-spore forming bacilli, and they ferment lactose with the production of gas and acid, and that's after 24 hours of incubation at 37 degrees Celsius. So as they grow and they use lactose for fermentation, the byproducts of that are gas and acid, so that helps us to detect them. So we can use those facts to run 3 basic sequential tests to detect coliform bacteria in water. Those 3 tests are called the presumptive test, the confirmed test, and the completed test. So the first test is the presumptive test, and that's testing for coliforms, just general total coliforms. It doesn't specify whether we have E. coli or total general coliforms. It's not a very specific test. So in this test, the test water is added to a red lactose broth containing an inverted glass vial, and that's known as a Durham tube. So what that means is within your test tube, there's another tiny glass tube that's turned upside down, and you can kind of see it in the picture here, so that if gas is produced by any bacteria in your sample, you'll see that gas trapped inside the inverted glass tube. So coliforms, again, are able to use lactose as their carbon source, and they produce gas and acid as a result. So in order to detect them, the acid changes the broth from red to yellow. So that's how we know if there's presence of acid. And then the gas production, if it's a coliform bacteria, the gas would be produced and it would become trapped in that Durham tube. So you would see it as a little bubble. So if you look at these pictures, the red tube is what you're starting out with, and when you add your sample and incubate it, if you have a sample with coliform bacteria in it, it would turn yellow, and you would see that gas bubble in the inverted tube. So if there's both yellow color and gas in any of the tubes, that's a positive test result. So that is the presumptive evidence of the presence of coliforms. So if your presumptive test suggests that the water is contaminated, now you have to move on to the next test to confirm it. Before we go on to the confirmed test, I want to just talk for a minute about most probable number, or MPN. So that presumptive test that we just did can also give an indication of the number of coliform bacteria present in the water. So it may be a small number, so not every tube from your water sample would be positive in that case. So that's called the most probable number, or MPN method. And this picture shows how it's done. So the first step is to perform the test with the Durham tubes using 10 milliliters, 1 milliliter, and 0.1 milliliter of water in 5 tubes each. And then you would incubate it and check your results. And in the picture here you can see from the 10 mLs of water we had 4 positive results. From the 1 ml of water we had 2 positive results. From the 0.1 ml they were all negative. So that number of positive tubes you use to consult the MPN index chart. And that gives you an MPN index or a range. And then you would check that healthy range and make sure it's a healthy range for whatever the purpose is of the water. So for drinking water, for example, would have to have an extremely low range or 0. And then, as it gets higher, it's acceptable for bathing and swimming. And then above a certain range, it's unfit and unhealthy. So from the presumptive test, you may have some tubes that were yellow and had gas produced that indicated coliform bacteria were present. So from that, you would move on to the confirmed test. And the presumptive test didn't really tell us what type of coliform bacteria. We couldn't specify if it was E. coli or not in that test. But the confirmed test is a little bit more specific. So the confirmed test uses selective or differential media such as EMB or eosin methylene blue to confirm your presumptive test. So how that works is methylene blue inhibits gram-positive growth. So anything that's gram-positive is not going to grow on these plates. But remember, coliforms are gram-negative, so they can potentially grow on these plates. And then the acid production, because remember that coliforms produce acid in the presence of lactose, that causes color changes on these EMB plates. So lactose fermenting bacteria like coliforms would appear as brown to blue-black looking colonies. But we can also distinguish E. coli specifically because E. coli appears as blue-black colonies with a green metallic sheen on them. And you can see that clearly in the picture here. And then the third test is the completed test, which is the final stage. And it checks the results of those first two tests, the presumptive test and the confirmed test. So what you would do is use a positive colony from the EMB plate and use that to go back and inoculate another lactose broth with the gas vial, the Durham tube, and you should see the production of gas and acid. So you're checking that again. And then you would inoculate an agar slant, which is a test tube with solidified agar in it, and you can grow the bacteria inside of that. So from that growth, you would gram stain the bacteria and analyze it with a microscope. Because remember that coliform bacteria you would see under the microscope are gram-negative bacilli. So those three tests are the standard practice for testing a water sample. But there's another separate modern method to test water called the Coliscan plate. So it's a separate test from the presumptive, confirmed, completed. It's simply another method. So those other tests required multiple steps to distinguish E. coli from the other bacteria and general total coliforms. But the Coliscan premise is that general coliforms produce galactosidase enzyme when they ferment lactose. But E. coli produces the galactosidase because it is a coliform, but it also produces glucuronidase. So there's chromogenic substances in the gel that get broken down depending on those two enzymes. So for general coliforms, the galactosidase production will trigger the production of pink colonies. And in E. coli, the combination of those two enzymes that E. coli produces, the galactosidase and glucuronidase, would trigger dark blue colonies. And then you might also see non-colored colonies that would be bacteria that are not coliforms. And Coliscan also contains inhibitors, so most other bacteria will not grow on them. So the advantage of this Coliscan plate method is that in one step you can determine if there are general coliforms present by seeing if you have pink colonies, but you can also determine if there's E. coli because they would produce blue colonies. So in one step you can determine what is present in your water sample.